You want to go live, yeah, dude.
Yes. So the only way to record it is this way. Okay.
One committee member, two committee members.
Sugar?
This is 3 to 5 days. It's not gonna kind of go up. Yeah.
That's not unusual.
Yeah. Apparently you can set it up so they can see it.
One, two, three. One more. Eric. I saw you. Eric. Of course.
Okay.
Write this on. There's still time to get coffee. So we can. Right. It's the. Yes. This is the downturn.
I really like it. Did you do it in that? Yeah, very much.
Aerospace engineering.
You're not expected. You're not expected. Well, French horn. That's funny.
Oh. You mean you've done committees and things that are not science?
That's from morning.
Okay.
Yeah, that's one way to.
Wow. Yeah, I'd like to do something.
Hi. Do you hear this?
Yeah, my impression is
Right.
you
Oh,
phrase
yeah.
Right.
Uh,
I think it would be a good place because I'll be moving back.
yeah, so they can see her.
The artist.
Because when
And yet you signed off on
like
it.
Well, you know, it's kind of awesome.
online somewhere.
All right, we'll get started. Thank you all for coming this morning to witness the culmination of Betsy's PhD program, journey to the PhD. I'm not sure that Betsy actually knew what cichlids were when she came to interview. She wasn't actually coming to interview for my lab, but what I knew about Betsy from her application was that she had a good head on her shoulders and that she had good hands in the lab and an interest in evolutionary genetics, so I worked hard to recruit her to the lab. During her first semester, I introduced her to what we knew about the diversity of sex chromosomes in cichlid fishes, and she soon latched onto the project she's going to talk about today, studying the beautiful anomaly that is B chromosomes. This required not only her skills at the bench, but also her penchant for logic and for a very methodical approach to hypothesis testing. Betsy's been an incredible asset to teaching genetics, my introductory genetics class. She earns high marks with the students while demanding their best efforts. She has also been the champion plagiarism sleuth, much to the dismay of a number of students. Her consistent and conscientious efforts as a TA earned her the 2018 Pritchard Teaching Award from the Biology Department. Betsy wrote a research proposal that won a $10,000 research award from the American Genetics Association. This award funded much of the RNA sequencing that she will discuss today. She's published 2 papers, one in Genes and the other in Journal of Heredity. There are 2 more chapters of her thesis that should be published in the next year. Her 2017 paper in the Journal of Heredity won the 2018 Stephen J. O'Brien Award for the best student paper in the journal. That was worth a $2,000 prize, as well as a $1,500 trip to Hawaii to present a paper at the annual meeting of the association. And if I can brag about the lab for a moment, Will Gammerdinger recently won the 2019 Stephen J. O'Brien Award for his paper on sex chromosomes. Go team. Betsy was also an invited speaker at an international chromosome conference in Brazil. So join the cichlid labs and see the world. Betsy has not yet decided where she will head next, but her work on B chromosomes has ignited an interest in the molecular basis of genomic conflicts. Wherever she goes next, she will be an asset and will make important contributions. So please welcome Betsy and her thesis on B chromosomes.
Thank you, Paul. So, if you haven't heard me talk about B chromosomes before, you might be asking yourself, what is B chromosome? Every species has a typical number of chromosomes in its genome. So this is a fish species known as a bleak. And the typical number of chromosomes in its genome is 25. That has 25 pairs of chromosomes. So this is a karyotype of an individual. Karyotype is simply an image showing the chromosomes in the genome. For some species, though, some individuals in those species have extra chromosomes. So this is a second individual from the same species, and it has this extra chromosome, which is referred to as a B chromosome. So the typical set, in this case the 25, are called A chromosomes, and this extra one is referred to as a B. You might have noticed that while all the other 25 are in pairs, this B chromosome is by itself, which is another key feature of B chromosomes. So looking at another example, this is a rocket deer from Brazil. And we have the karyotypes of 3 individuals, 1, 2, and 3. And if you look at the B chromosomes in these 3 individuals, the first one has 2, the second has 4, and the third one has 6. So B chromosomes can vary from cell to cell within an individual, and more commonly from individual to individual, where individuals have the same number of B chromosomes in every cell in their body. So if we start to form a definition then, B chromosomes are not found in every individual of a population or species, which makes them non-essential. You can have normal, healthy individuals that lack that chromosome. They vary in number per cell. You are probably all familiar with trisomies, the common example being the trisomy of chromosome 21 that leads to Down syndrome, where essentially you have a third copy of one of the A chromosomes. And I want to make sure that you understand that A chromosomes are not simply an extra copy of one of the A chromosomes. They are unique compared to the other chromosomes. However, they do share sequence with the other A chromosomes in the genome. It's been found in several species now that B chromosomes have sequences from most or all of the A chromosomes. Once those sequences are on the B chromosome, they often undergo a series of duplications, making B chromosomes very repetitive. So they have the same sequence copied multiple times. And the reason why this is important is repetitive sequence is very hard to assemble. So in the era of genome assemblies, there's actually not a single complete B chromosome assembly or anything nearing complete, despite having found B chromosomes in so many species. Another important feature of B chromosomes is a process called drive. So drive is anything that increases the transmission of a genetic element to the next generation. If you imagine you have a gene with 2 alleles, according to Mendel, the offspring of that individual should have a 50-50 chance of having the first allele or the second allele. So any mechanism that transmits one of those alleles more than 50% of the time would be a mechanism of drive. B chromosomes have multiple opportunities for drive. So we think about the cell divisions a eukaryotic individual undergoes, and we consider one pair of homologous chromosomes. That pair of chromosomes is going to replicate before mitosis, and the newly replicated copy, the sister chromatid, is going to separate in mitosis. Going to replicate again before meiosis 1. In meiosis 1, the homologous chromosomes are going to separate, and then meiosis 2, again, the sister chromosomes are going to separate. Each one of these divisions is an opportunity for B chromosomes to manipulate some process of cell division in order to increase its transmission or to drive, along with fertilization. And we'll talk more about possible drive mechanisms. So I've given you examples of B chromosomes found in mammal species and fish. Another fish species you might recognize would be sea bass. These are known to have B chromosomes. Looking at other mammals, the Korean field mouse and this really cute guy called raccoon dog. These both have B chromosomes, along with the raccoon dog's close relative, the red fox, also a species you might be familiar with. Among other vertebrates, we see reptiles as a microteid lizard that has B chromosomes. And beyond vertebrates, so in homage to invertebrates, grasshoppers and fruit flies are very well studied systems with B chromosomes. For the plant people in the room, corn and rye are probably the best studied systems for B chromosomes, and we also find them in species like the Mediterranean hyson. And you can even find them in fungal species. So what I'm trying to convey here is that B chromosomes are very common in eukaryotes. They're actually present in about 15% of all eukaryotes. But if you were to look at the individuals from any one of these species, you'd be hard pressed to look at the individuals and be able to tell which one carries a B chromosome and which ones don't. They don't typically have very many associated phenotypes. An exception to this would be the corn plant. So here we have the stalk, here we have a close-up of the leaf, and the phenotype that is associated with this B chromosome is a striking pattern. But this phenotype actually only emerges when the individual has at least 4 B chromosomes, and other phenotypes like reduced fertility and zigzagging of the stalk and reduced vigor only show up when they have more than 10 B chromosomes. So even in the cases where we can find phenotypes associated with that B, you have to have many B chromosomes before you start seeing those phenotypes. So back to our definition, few associated phenotypes. And so because we don't have an assembly of the sequence, the chromosome sequence has been hard to examine in the past, and because there's so few associated phenotypes, the historical view of B chromosomes is that they're inert. They really don't do much. They have some sequence on them that is controlling drive, but other than that, it's mostly junk sequence, junk DNA that doesn't do anything. In contrast to that historical view, in the last few years, there have been wonderful improvements in next generation sequencing, and as a result, a surprising number of genes on B chromosomes have been identified, and even some evidence of transcriptional activity of those genes, which is starting to contradict the historical view and suggest that chromosomes might actually have perhaps cryptic phenotypes that we weren't previously appreciating. So I've been studying B chromosomes in African cichlids. Cichlids are a family of fish. Shown here in Africa, there are 3 rift lakes, Lake Victoria, Lake Tanganyika, and Lake Malawi. And these are just some of the examples of the cichlids that live in those lakes. They're a wonderful study system for speciation and evolutionary genetics. Shortly before I began my thesis, our colleagues from Brazil, this is Dr. Cesar Martins and his former student Irani, who's actually here at the university now, and Andrea, were able to identify B chromosomes in some of these cichlid species. So in a single species from Lake Victoria, Statotilapia latifasciata, they found males and females that harbored B chromosomes, and these individuals could have 1, 2, or 3 B chromosomes. Since then, several additional Lake Victoria cichlid species have been shown to have Bs. In fact, every single species examined was shown to have a B. But the work they did also identified a single species from Lake Malawi. This is Metroclima lombardoii. This is the species I'll be focusing on today. They found karyotype data suggesting that these fish also have B chromosomes. Focusing on that Metroclima lombardoii from Lake Malawi, the females of this species are usually bright blue, and the males are bright yellow. I've shown here a karyotype from a female that's lacking a B, a karyotype from a male lacking a B, and a female that has the B. One of the interesting things about their results was that of the 9 individuals they were able to show with karyotype data that had B chromosomes, all 9 only had a single B, in contrast to the Lake Victoria cichlid species that had 1, 2, or 3. And all 9 of those individuals were female. So this is where I came in with my list of questions to start my thesis. The first question for this newly discovered B chromosome is whether or not it follows suit for the majority of B chromosomes and whether it experienced drive. If it did experience drive, how did it accomplish that? Since we see a variety of mechanisms in chromosome systems. I was also very interested in the fact that all 9 individuals that they had karyotype data for were female. I wanted to know if this was the result of a small sample size, and with increased sampling, we might start to see individuals with 1, 2, or 3 B chromosomes, as well as in males, as you see in Lake Victoria cichlid species, or if this was indicating something about the biology of that B chromosome, and these B chromosomes are restricted to females in the Lake Malawi cichlids. Also, in light of the recent changes in the perception of B chromosome activity, I wanted to examine the genes on the B chromosome as well as their transcriptional activity, and was able to do this with the really wonderful resources we have, the genomic resources we have for African cichlids. So to start out, I needed to identify additional individuals with B chromosomes. I didn't have access to karyotype data. I only had access to genomic samples. So I used PCR to genotype for presence or absence of a B chromosome. This was possible because of work by my colleague, Conti. where he had an idea of sequences that might be on the B chromosome. So using those karyotype individuals, I confirmed that those sequences were on the B chromosome through designing primers and showing that they only amplified in the karyotype individuals with Bs as opposed to the karyotype individuals without Bs. And use those primers to investigate more samples. So not only did I look in additional individuals from metroclimal Lombardi, but I expanded my search to other species from Lake Malawi. And I did so in both females and males. For the females, we found not only additional B individuals from metroclimal Lombardi, but also in 6 other species from Lake Malawi. And all 7 of those species didn't find a single male for B chromosome after looking at over 300 individuals. So confirming the B chromosome of Lake Malawi cichlids really is restricted to females. It was not a result of a small sample size. The other question I had from the karyotype data was about the number of the B chromosomes. So again, like Victoria, they had 1, 2, or 3. Those 9 karyotype individuals only had 1. This is a little bit more difficult to do with genomic data. So imagine this is our B chromosome. I told you that the B chromosome has repetitive sequences. So imagine each color is representing a different type of sequence. And then, so black is one type of sequence. It has 2 copies on the B, blue is 3, so on and so forth. Ideally, you would be able to use qPCR to quantify the copy number of that sequence, and from that determine the number of chromosomes. Because B chromosomes are so repetitive, they're prone to duplication and deletion. So any 2 B chromosomes might not have the exact same number of copies of each of these sequences. So to get around this, I actually looked at 5 separate sequences on the B chromosome with the idea that if you have a second chromosome, then on average, across those 5 sequences, you should see generally a 2-fold increase in copy number. After looking at this for the 43 B individuals in those 7 different species, we did not find evidence of a single individual with 2 or 3 or more B chromosomes. So all of the individuals only ever had a single B chromosome, if they had a B chromosome at all. The reason why this is important is it tells us something about their drive mechanism. So if you imagine that lonely B chromosome that doesn't have a homologous pair can replicate normally, and it can go through mitosis normally. But as it prepares to go through meiosis 1, it has a bit more of an issue. It can either go to one of the resulting daughter cells or the other because it doesn't have a homologous chromosome to segregate. There's only one. The B chromosome is limited to females, and female meiosis is asymmetrical. So of these 2 daughter cells, one of them is called a polar body and is not going to produce an egg that gets passed on to the next generation. It's going to actually disintegrate. So if the B chromosome goes to this cell, it's not transmitted. If it goes to this cell, it is transmitted. Which leads us to our hypothesis of drive. If this B chromosome manipulates meiosis 1 such that it more often goes to this other cell, which is called the egg generative nucleus, then it'll be more often passed on to the next generation, so above Mendelian expectations more than 50% of the time. Now that we have an idea of how the B chromosome might be driving, I need to make sure that it is driving. This can be examined with a simple genetic cross. So I took my male individuals, which of course do not have B chromosomes, and females that either do or do not have B chromosomes. Really great thing about Lake Malawi cichlids is that they're mouth brooders. This is not the same species, but it's a great image to demonstrate this. If you see these little black dots in this female's mouth, those are actually the eyes of her offspring. So after fertilization, she scoops up her offspring and she holds them in her mouth for the next 2 weeks to give them the best chance, the best start in life. And it's easy to collect this female and take those offspring from her so that they can be reared together as a family. And once they reach sexual maturity, I can then collect information on them. So again, our alternate hypotheses, the B chromosome is not driving, so it has equal chance of being segregated to the egg generative nucleus and being passed on, or to the polar body and being lost. Or we do have drive. This B chromosome is preferentially segregating to the egg generative nucleus. It's choosing that daughter cell, so it's more often being passed on to the next generation. So more than 50% of the offspring will have a B chromosome. So I was able to collect data for 6 different families. And we actually found that B chromosome transmission rate varied a lot. So everywhere from 50% all the way up to 95%. There's 2 possible explanations for this variation. One is sample size. So if you look at the extremes of this range, the 50% and the 95%, these were actually my 2 smallest families, so the families with the least number of individuals in them. So it's possible that if I had larger sample sizes, the variation in this transmission rate might decrease. Another explanation, however, is that this variation represents the B chromosome's ability to drive. So a bee transmitting itself more frequently is beneficial to that chromosome. It's not necessarily beneficial to the rest of the genome. And so you'd expect the rest of the genome to evolve mechanisms to suppress that drive and resulting in an evolutionary arms race. So this variation could be the result of different B chromosomes in different A genome backgrounds having a different opportunity for drive. And it would be really wonderful to collect additional families from each of these couples. So the couples that had produced these families here, to see if every time they reproduce, they have the same transmission rate when you have the same B chromosome in the background A genome. So the first question was, does the B chromosome drive and how? It does drive, and we have the hypothesis that it drives during meiosis 1 using preferential segregation. Second question was, is it really only in females? And if so, why? I showed you with population data that it really was restricted to females, so that leaves us with why. I have 4 hypotheses. The first one is elimination of B chromosomes in males. So we see something similar in nature in the jewel wasp. The jewel wasp has a B chromosome and it's haploid diploid. This means that when the female lays an egg, if that egg is fertilized, it now has 2 copies of every chromosome in the genome and it'll become female. If the egg is not fertilized, it only has 1 copy of every chromosome in the genome and it becomes male. The B chromosome carried by these males actually alter the chromatin structure of the paternal chromosomes, the chromosomes inherited from the father, shortly after fertilization, so that in the first few mitotic divisions, they don't go through mitosis properly and they actually get eliminated. So the B chromosome in this jewel wasp eliminates the paternal chromosomes from the genome, resulting in a haploid individual with the B chromosome that becomes male. So I'm suggesting the opposite. Instead of the B eliminating the paternal chromosomes, I'm suggesting something about the male genome might eliminate the B chromosome. Another opportunity would be segregation with a W chromosome. So if you imagine we have our X and our Y sex chromosomes. They pair during meiosis and segregate to different gametes. The gamete that gets the X will produce a female individual, and the gamete that produces the Y, or that inherits the Y, will produce a male individual. In a species of insects, we have a B chromosome that is known to pair with the sex chromosomes, so that it's partially pairing with the X, and when they segregate, that B goes to the same gamete as the Y, and so it's always passed down to a male. This doesn't quite work for our cichlids because they're only found in females, not males. But if we flip this from an XY sex determination system to a ZW, so WZ is female, ZZ is male, we have a B chromosome that pairs with these sex chromosomes such that it's partially pairing with the Z and segregating the same gamete with that W. Because the W determines femaleness, this gamete would produce a female, and that B chromosome will only ever be inherited by females. Third hypothesis is that the B chromosome is acting as a sex chromosome itself. So you can imagine if we have an XY sex determination system, XX is female, XY is male, And you can either have those genotypes with or without the B chromosome. But if that B chromosome is acting as a sex chromosome that feminizes, then it would convert these XY individuals to females. So I'm denoting females with pink, males with blue. And the last hypothesis is male lethality. So this is the idea that males and females can inherit the B chromosome, but if you're a male with the B, that's not a good combination and you're not going to make it very well. So again, these would be the different genotypes you could have, but if you're XYB, this would be lethal, denoted by gray. So if we look at these 4 hypotheses, we can actually put them into 2 different categories. The last 2 alter the sex ratio. So if you're converting males to females, or if you're killing males, but not females, then that would change the sex ratio, whereas our first 2 hypotheses didn't. So looking for differences in the sex ratio is a really simple thing we can examine with genetic crosses. So comparing B chromosome families, no B chromosome families to B chromosome families. In this plot, the y-axis is going to be the percent males in those families. In the families that lack a B, we see a median sex ratio of about 45% males, or a mean of about 48% males. Again, the 2 hypotheses that would alter the sex ratio both predict that we would see a decrease in the frequency of males. And that's exactly what we do see. So going back to our hypotheses, we can eliminate the first 2 because we do see a change in sex ratio, and we can just focus on those last 2 hypotheses. To distinguish between these 2, I did a linkage analysis. So this tends to be a little complicated, and we'll walk you guys through step by step. So up here I have representation of the mother of this family and the father of this family. This is specifically a family lacking B chromosomes. And I looked at markers, so these are microsatellite markers along linkage group 7. Linkage group 7 is known to have an XY sex determination system, and we had reason to suspect that metroclimal lombardoi, which is what this cross was done with, had the XY sex determination system on linkage group 7. So this is the first place I looked. The numbers are the alleles, but you can actually ignore that. I would focus on the color. Essentially, these are the alleles inherited from the mother. You can have the pink, the light pink, or the dark pink. These alleles were on one chromosome, and these alleles were on the other. So this is essentially one chromosome you can inherit from the mother, the other chromosome you can inherit from the mother, one chromosome you can inherit from the father, and the other you can inherit from the father. Looking at their offspring, so in this table, each row is an individual produced from this cross. On the top, we have the females. On the bottom, we have the males. And the pattern we see is that all of the females inherited one of the chromosomes from the father, and all of the males inherited the other chromosome from the father. The reason for this is that this chromosome is actually the X chromosome, exactly what we expected. While the chromosome inherited by the males is the Y. So if you inherit the X from the father, if you're female, you inherit the Y, you're male. And it doesn't matter which chromosome they got from the mother here because they're both X chromosomes, they don't determine sex. We do the same thing with a B chromosome family. So again, I'm just showing 2 markers here. These are the chromosomes inherited from the mother, the father. These are the offspring. On the top here are all the individuals with B chromosomes, and on the bottom are the individuals without separated females and males. If we just look at the individuals without B chromosomes, we see the same pattern. The females have the X, the males have the Y. But when we look at the individuals with B chromosomes, they can have both the X and the Y from the father, and yet they're still female. The reason for this is that the B chromosome is acting as a sex determiner. It feminizes the XY individuals, making them female. It's an epistat dominant sex determiner that feminizes. So I can't necessarily rule out that if a B chromosome is in a male, it would be a lethal combination, but I have positive evidence that it is a sex determiner or a sex chromosome. So moving on to our next question, what genes are on the B chromosome? I turn to a genomic analysis. So instead of just looking at Metroclima lombardi, here I looked at all 7 species. I took genomic DNA and sequenced these mostly with Illumina data, or Illumina sequencing, which produces these short reads, which can then be aligned to a reference genome. So imagine we're looking at 2 different individuals. The black bar here is symbolizing that reference genome. We have an individual that lacks a B and an individual with a B. The reads produced from their A chromosomes should align to that reference the same between these 2 samples. However, the reads produced from the B chromosome don't have a reference to align to, right? There's no reference assembly for the B chromosome. Remember, I told you that the B chromosome has sequences from all of the other A chromosomes. So if you imagine this orange bar here is a portion of the A chromosome that also has copies on the B chromosome, so homologous sequence, then the reads produced from the B chromosome, if the sequence is still similar enough, could align to that portion of the A genome, producing an increase in coverage or read depth. The comparison of coverage between these 2 samples is called a coverage ratio analysis. So what I'm showing here, this is going to be data from Labiotrophus truaviceae. So Labiotrophus truaviceae has a population that doesn't have B chromosomes. So this is male genomic data without B chromosomes and female genomic data without B chromosomes. What's being shown here is this bar is representing linkage group 9. The red is denoting what part of that linkage group we're looking at. So this space here is this part of the chromosome. And in gray, you have the coverage, the number of reads that are aligned to that position of the genome. In the vertical colored bars, those are just SNPs or mutational differences between the sample data and the reference genome. For these for the 2 samples, the males and females without B chromosomes, you don't really see much of a difference in the read coverage. But when we look at a Labiotrophus truaviceae population that does have B chromosomes, so this is male genomic data, and this is a female individual with a B chromosome, we see that increase in coverage, which was properly identified by our bioinformatics analysis. And I'm going to be referring to these regions that have this higher coverage and therefore a copy on the B chromosome as B blocks. So if we look at where these B blocks are, sorry, I forgot to give you guys a second example. So this is just another example of a B block to demonstrate that not all of them are continuous, some of them are fragmented. This could be because of an insertion in the A genome or a deletion in the B. Again, it's just to emphasize that the B blocks can be fragmented. If we look at where these B chromosomes are in the genome, I'm going to walk through an example for one linkage group and then show you the rest of the genome. So in this case, linkage groups are almost synonymous with chromosomes. We have the horizontal bar denoting that linkage group with the numbers in megabases. If we know where the centromere is for that linkage group, it'll be denoted with a yellow circle. And then the B blocks are shown in vertical black bars. Above and below those black bars are bar graphs. So the red bars above represent Metroclima lombardi, and it's the copy number of those sequences. So here we have the position of the B block, and then the estimated copy number of that block in Metroclima lombardi. And then below in blue is Labiotrophus truaviceae. The reason I chose these 2 individuals out of the 7 species is simply that they're in different genera. So looking across the entire genome now, we see that these B blocks map to every single chromosome in the genome. So much like what was seen in other species, sequence from the B really is similar to sequence on all of the other A chromosomes. So just looking at the Metroclima lombardi individuals, I identified 10 to 14 megabases of these B blocks. So if you sum the length of all of these B blocks from the reference genome, about 10 to 14 megabases. I then took those blocks identified in each of the 7 species and required that it be shared, so all 7 species have to have that block. I have 1.37 megabases. Again, this is shared sequence, shared B blocks across those 7 species, and I'm going to be referring to this as the core blocks or core B chromosome sequence. Taking that core sequence for the mesenchymal lombardois individuals and multiplying each block by its estimated copy number, I've captured an estimate of 24 megabases of the B chromosome. Going back to the karyotype data, this is mesenchymal lombardois, this is the B chromosome. So using the genome size and a comparison of the chromosome size, we can estimate that this B chromosome is about 15 megabases. So the core sequence of these chromosomes is roughly half of the B chromosome we expected. In this core sequence are 144 genes, only 14 of which are complete. So partial means that only part of that gene was transferred or copied onto the B chromosome. I do want to emphasize, though, complete doesn't mean functional. It just means that the entire gene was copied onto the B chromosome. Once on the B, it might have acquired mutations and bits and genes. So are there genes on the B chromosome? Yes, there's 144, though most of them are fragmented. So now I wanted to look at the transcriptional activity of going back to just mesenchymal lombardois, I took 3 females with the B chromosome and 3 females without a B chromosome. From them, I took samples from brain, liver, and gonad, ovary, since they're females, and extracted RNA, so not DNA, but RNA. Again, I used Illumina sequencing and aligned this back to the reference genome. So this time I had both the reference genome assembly as well as a de novo transcriptome assembly that I assembled from the RNA data. I was able to do a differential expression analysis, which means that I looked for genes that were significantly differentially expressed between B and no B females. In brain, I identified 1,266 significantly differentially expressed genes. In liver, 6,352, and then only 540 in gonad. So these are heat maps demonstrating that differential expression. The columns represent individuals, so we have our 3 B individuals to the left and our 3 no B individuals to the right, and it's the same order for each of the 3 heat maps. Each row is a different gene that's differentially expressed. The more yellow the coloration for that gene, the higher it is expressed. This is relative expression. And then the more orange-red, the lower the expression. So looking at the heat map for brain, you can see that more than half of the genes have lower expression in the B individuals than the individuals lacking it, which is also a pattern you see for gonad. Liver's a little bit more. Taking these significantly differentially expressed genes, I wanted to look at what might be causing it. So you can argue that differential expression is coming from sequences transcribed from the B itself. The B has 144 genes, it's transcribing these genes, and this is leading to the differential expression. Alternatively, you can have a copy of a gene on a B. It doesn't have to be transcribed, but simply having those extra copies alters the copy that's on the A chromosome. So maybe even though it's not transcribed, it still has binding sites for something like a transcription factor. Those transcription factors bind to the B copy, decreasing their binding to the A copy and result in a decrease of expression of that A gene. And the third option is that something about this B chromosome is changing the transcription of the A gene without having a copy of that A gene on the B. So it's not being transcribed from the B, there's not a copy of it on the B, and yet it's still affecting the transcription of the A gene. Looking in brain, of the 1,266 differentially expressed genes, only 10 of them even have copies on the B chromosome at all, leaving the other 1,256 to fall into that third category. In gonad, we see the same pattern, 14 on the B, the majority in the third category. And again, the same thing in the liver, which is suggesting that something about this B chromosome being present in the genome is altering the expression of A genes. I mentioned with the heat maps that in brain and gonad, more than half, so about 65% of the genes are upregulated in no B individuals and have lower expression in B individuals. But if we look at the genes of the 144 genes on the B, the ones that are differentially expressed, 90% of them have increased expression in B individuals. So despite the genes that are actually on the B chromosome having increased expression, we still have a general trend of a decrease in expression in B individuals, suggesting that B might globally cause a decrease in expression of the A genome, perhaps through something like chromatin remodeling. Now that I know which genes are on the B, I can look for B-specific expression from those genes. And I can do this with SNPs. So this is, again, a coverage plot. This time, it's of a metroclimal lombardi samples. So we have the male genome data, and this is a female with the B chromosome. I've altered the scales so to basically raise this up so you can see. So the scale here is 100, the Y scale here is 320. But the point here is that in this B block region, the female with the B has SNPs that are absent from the male genomic data. And the reason for this is that these SNPs are actually on the copies from the B chromosome. So by looking for SNPs within B blocks that are present in the female with the B and absent from the males without the B, we can identify B-specific SNPs, which are shown up here. We can do a bioinformatic analysis with these SNPs, take those RNA reads and sort them as whether they came from the A chromosome, the B chromosome, or if they don't contain any SNPs, and we can't tell. So these were our three possibilities explaining differential expression. We focus on that top one, the ones transcribed from the B using those SNPs. I found 46 genes in the brain, 50 in gonad, and 36 in liver that are expressed from the B chromosome itself, which is a total of 53 genes that are expressed in at least one of these three tissues. It's a lot more than you would have expected based on the historical view of the chromosomes. We also have those genes that don't have SNPs to sort. These are interesting because they either suggest that that gene has very recently found its way to the B chromosome and hasn't had a chance to acquire those SNPs, or it's been on the B for a while and it's doing something beneficial to the B, and so any SNPs that are start to accumulate are removed with purifying selection. So that makes these potentially interesting, and there are about 65 of them that are expressed but don't have any SNPs to sort. So coming back to our last question, is the B chromosome transcriptionally active? Yes, it's definitely not inert. There are 53 genes that are expressed from the B and an additional 65 that could be, we're not sure, and we've shown that the B chromosome affects the transcription of the A chromosome sequence. I'm going to take a couple minutes to talk about some candidate genes that have arisen from this. I'm going to focus on just three: centromere protein E, or CNPE, mitotic arrest deficient 2A, MAD2A, which is a mitotic spindle assembly check protein, and intercentromere protein, or INSENPE. For CNPE, I've been able to show that this is partially on the B chromosome. It is missing several exons in the middle of the gene. It has differential expression in gonads such that the B chromosome individuals have higher expression, though it's not significant. It has a p-value of less than 0.05, but it just missed the significance threshold when you account for multiple tests. And I've identified the specific transcription for this gene, so it is being transcribed from the B chromosome, even though it's missing those exons. For MAD2A, again, it's on the B chromosome. It looks like it might only be present in one or two copies, but it looks like that copy could be complete. It has differential expression in brain, so not in gonad, but in brain. Again, higher expression in the individuals. Again, not quite significant expression though, or significant differential expression. And again, I've identified the specific transcription. And for INSENPE, this is also only partially on the B chromosome. The opposite of CNPE, only the middle portion of this gene and its first exon are on the B. It has significant differential expression in the gonad such that the Bs have higher expression, B individuals have higher expression, and we see expression from the B chromosome itself. So what do these genes do, or what do the proteins they produce do? So we think about our homologous chromosomes in meiosis 1. This is simplified. Homologous chromosomes in meiosis 1 should have sister chromatids, but I'm trying to go simple here. They'll have a kinetochore associated with the centromere and a microtubule that'll bind to that kinetochore to pull those chromosomes apart during cell division. We focus on this region here. SNPE is a kinesin-like motor protein that one half of it binds to that kinetochore, and the other half binds to a microtubule. And the side that binds to the microtubule is actually the motor portion of this gene, and it actually helps to move that chromosome during cell division. So there are other examples of kinesin-like motor proteins being involved in meiotic drive, not necessarily for B chromosomes, but for other chromosomes, with the idea that something about this protein, if there's an alteration of this protein that makes it faster or stronger as a motor, then it might be able to pull that chromosome to the better side of the cell during division that will lead to the agenic nucleus. We also have MAD2A and INSEN-P, both of these associated with the kinetochore. Interestingly, MAD2A is recruited to the kinetochore by SNPE, and both of these act on the mitotic checkpoint complex. So it says mitotic, it's also important for meiosis. It's just first studied in mitosis. But this mitotic checkpoint complex, depending on its state, will either suppress or initiate anaphase. Anaphase is the separation of those chromosomes. What I haven't told you about B chromosomes is that they often lag through cell division. So they've been shown in several species to be the last chromosome to complete DNA replication. And they've been shown in several species to have a hard time segregating to either nucleus during meiosis. There was actually a study with an insect species where individuals had 2, 4, or 6 B chromosomes. When they had 2 B chromosomes, they segregated fine. When they had 4, they were very slow to get to either side. And when they had 6, they were so slow that when the nuclei reformed, the B chromosomes were left in the cytoplasm and lost. So lagging through cell division is a problem for B chromosomes. And you can imagine if the alteration of these proteins could change the state of the MCC so that it's suppressing anaphase or delaying anaphase to give the B chromosome enough time to get it to a more favorable position during cell division so that it can be segregated to the agenic nucleus. So it's all a little bit hand-wavy, but these are the genes that I'm most excited about for candidate genes for drive. Going back to my original questions, does this B chromosome drive? Yes, it does, with a lot of variation. We think during meiosis 1 using preferential segregation, and we have 3 candidate genes that might be underlying that behavior. Is the B chromosome limited to females, and why? It is, and the reason is because it's an epistatically dominant sex determiner that feminizes. What genes are on the B chromosome? We've identified 144. Again, most of these are fragments. And is the B chromosome transcriptionally active? Yes, it is. It is not inert. And we've also shown that the B chromosome affects the transcription of the A genome. So looking forward, I think cichlids are a really wonderful system for studying B chromosomes. And while I've talked to you about a few of our candidate genes for drive, I didn't mention anything for the sex determination. The only thing I've actually come across is a gene called NANOS2. The reason why I'm not super excited about this is this gene is actually involved in male development. So this is after the male fate has been decided. NANOS2 is important for launching male development. You could imagine that a non-functional copy of NANOS2 might then reverse that decision. So when it's trying to develop as a male, it fails to do so, reverses, and develops as a female. I feel like this is probably a little bit more hand-wavy than my previous explanation. But there's still a lot of work to be done to identify how this B chromosome is determining femaleness. For the candidates I've identified for drive, there needs to be several studies to demonstrate functionality, not just that these are the genes underlying drive, but how they work on a molecular level. For my thesis, I've identified a large portion of the sequence on the B chromosome, but we don't have any idea of the structure, the orientation of those sequences, what's next to what, whether these repeats are tandem or interspersed. And I think with the developing technologies, especially long-read technologies like PacBio, in the next few years, we're going to start seeing these assemblies of B chromosomes. And then I think it would also be very interesting to examine how the B chromosome interacts with other sex chromosomes. So cichlids are really amazing in their diversity of sex chromosomes. In Lake Malawi, including the B chromosome now, we have at least 5 different sex determination systems, and in closely related cichlids, we have more than double that. So looking at how the B chromosome interacts with these other sex determination systems, since these closely related species are easy to hybridize, might tell us something about the regulation network, the genetic network of the sex chromosomes. With that, I would like to thank you guys for listening to me talk about B chromosomes for an hour. I'd like to thank my committee, especially my advisor, Tom Coker. When I made the decision to go to grad school, I was working as a technician in a lab, and I loved bench work and knew I could be happy doing that forever. And the reason why I decided to go to grad school was that I wanted to learn, I wanted to be the person to ask the questions and figure out how to answer them. And I wanted to learn how best to do that. I can't imagine learning from anyone better than Tom. I'd also like to thank Cesar Martin's lab from Brazil. This is an image with some of his current students, but I also like to thank his past students, Irani, Andrea, and Diego. The work they did before I started my thesis was really my diving board into this research. I actually didn't know B chromosomes existed until I heard Cesar come visit the lab and give a talk, and I've been enraptured with B chromosomes ever since. I'd like to thank the cichlid labs here at the university. These are people that I've worked with for the last few years and had a whole lot of fun with, and their support in talking about science, as well as their friendship, has meant a lot to me. And finally, I'd like to thank my husband and my family for their tremendous amount of support, including never questioning my crazy decision to go to grad school in the first place. With that, anybody have any questions?
Super cool. So I'm kind of curious about, I guess, how similar these genes are and how differentially expressed they actually are between the two, like the no B and the B individuals. Like, are there some that are just like really, really different? Or are they all kind of similar? Like, what's the...
I knew if I took this out, you would ask. So I'm going to have to take a minute to find the volcano plots. So a volcano plot is just showing you on the x-axis, this is fold change on a log scale. So this is saying compared to the B chromosome individual, was the no B chromosome individual, did it have higher expression or lower expression? And then the y-axis is the log scale of the p-value significance. Everything in red is significant. Everything in black doesn't meet that significant threshold, and that's after correcting for multiple tests. And so the further you are away from that middle line, the more dramatic the differential expression. And we definitely do have some that are both very statistically significant and very differentially expressed. There were also several that I was looking at, I can't remember which was which off the top of my head, but several that had very, very high FPKM. So FPKM was the measure of the expression. Some of them were significant, but the expression was, say, less than 50 FPKM. And we had some that were well into the thousands.
I mean, so for me, like, how do you sort of reconcile that with this idea that there doesn't seem to be a very huge phenotypic difference? Is it just that we're not looking at the right things phenotypically?
I think that's what it is. I think it's cryptic phenotype. So I think that if it's interfering with something that leads to too high of a mortality rate in the individual, so essentially these B chromosomes are selfish, so their A genome is their host, and if any parasite that is too detrimental to the host doesn't last very long, and so I think it is causing changes, but it's not the changes that would result in something very wrong going on.
So you said you found 14 complete genes on these B blocks, and but you found a lot more genes that differentially expressed, a lot more than 14. So are these partial genes being expressed as partial transcripts, or they're just lack of knowledge of completeness?
Here we go. I never actually put the other one up for that. So this is one of my candidate genes, NPE. At the top, I've got the coverage. In green, I've got, it's similar to core blocks, but it's just what's shared in the Lombardo-White individuals that I looked at. Orange is a gene annotation, and purple is the de novo transcripts that have been aligned back to the genome reference. And so for SNPE, in the gene annotation, this and this were identified as SNPE. So this was identified as SNPE, and this was supposed to be SNPE-like, but we have transcripts that span this entire region, so I think that was just an issue with the annotation. The B blocks cover the second half and the first half of this gene, and we have transcripts that have the exons here missing the ones in the middle and the exons on the other side. So the partial genes essentially can be transcribed from the B. These are producing truncated proteins. Most of them probably are just pseudogenes that are being transcribed, but you can have truncated genes that possibly do something like interfere with the function of the original protein. So if this is a truncated protein, but it still has enough to partially do its job, that semi-functional protein is getting in the way of the functional protein being produced from the A genome, and it could be interfering with whatever that process is.
In your last slide, you said B stands for best.
Yes.
And you also mentioned early on in the introduction that about 15% of genetic genomes are known to have Bs.
It's a really soft ballpark because most of the B chromosomes that have been discovered were completely accidentally. So
that seems like a minimum. And what I'm curious about is how, in typical sequencing, when you see copy number variation, but when you compare individuals, is there any way to easily determine whether that could be on a B chromosome? I mean, are there potentially way more B chromosomes floating out there?
yeah, so with the late allowing syphilis, we had the benefit if they were only female. So you have male and female data, you notice these regions of increased coverage. B chromosomes are in both males and females, and you're just having the same population data, you're just going to notice regions with higher or lower coverage that maybe is repetitive. Matt Conti actually found out after the genome assembly of one of the other syphilis species was completed that the individual they used to produce those sequences had B chromosomes. And it had led to a lot of misassemblies in that reference assembly, which is the reason why we usually suggest that. I get the feeling people are starting to use cytogenetics as maybe outdated technologies, and genomics is the new hot thing. Genomics is really wonderful. It has a lot of tools to answer questions that we haven't been able to answer before. Cytogenetics is still very useful, and it's probably a good idea to karyotype individuals before launching into a genome project. Yeah.
I realize I'll have an opportunity to ask one question, but this one, I can't help but ask. So you have not mentioned transposable elements at all in anything that you've said. And
No.
I guess I'm curious, if you were to indicate where transposable elements are in the genome, are they more often in the B blocks?
I haven't quantified that. I definitely have come across a lot of transposable elements in the middle of B blocks, on the edges of B blocks. I think there's, a couple PhD theses there to be done on transposable elements and B chromosomes. The thing I'm most interested in would be how sequences get copied onto the B chromosome. The most common theory is transposable elements excising themselves and moving them to the B chromosome. There's definitely, I've come across a lot of transposable elements, but I haven't done any quantitative work to be able to.
And I guess so. Has anyone looked, or have you looked if there's evidence of retrotransposons? So there's lack of introns in B blocks.
Yes, we actually found that. So most of my samples were sequenced with Illumina. I have the reference, the individual used to produce the reference was sequenced with PacBio, and I have a single metrochromosome Lombardi individual with the B chromosome that was sequenced with PacBio. And we found by comparing metrochromosome Lombardi individuals that were sequenced with Illumina and with PacBio that the reads aligned differently. So there were some regions that looked like a retroinsertion in one type of data and not in the other, whereas there was actual retroinsertions that were completely missed by. So the Illumina data, because it can align shorter reads, was aligning exons that were very highly similar, but it was failing to align the very different introns. So it looked like a retroinsertion, but when we looked at the PacBio data, it aligned the entire thing and we had a complete block, whereas the PacBio data was able to capture some of the actual retroinsertions that were missed. So.
Okay, the committee's gonna go ask our questions for a couple of hours. Assuming that goes well, I expect there will be some celebration and refreshments on the second floor of the